Good afternoon, everyone. Today we are going to have a live session of the Proteomics course. I hope you have all gone through all the videos of the week one, and we'll wait for 5 minutes if anyone else wants to join, and then we can start. Yeah, I'll just share my presentation. Let me know if it is visible. Is the presentation visible?
Yes, Neha. Sneha, are you facing any issue?
No, no, I was just waiting if anyone else wants to join because once I start presenting, I mean, I don't know if anyone has joined. So,
Uh, can you make me host or something that I can admit?
sorry.
Like, can I admit people? Like, only you can do it, or if you can make me
Just a
host,
second.
I can.
the host country. Yeah, I'm not able to figure out currently.
Okay, it's okay. You can start after 2 3 minutes. Sneha, can you go to people? There, you will see three dots, and there you can make me co-host. Just
Okay.
go once in the people list.
In the people yeah, where my name is mentioned, let me
Yeah, in my name, you can click on the three dots. Then I think the co-host option will come.
okay, but it's just showing remove from the
Okay, okay, no issue. You can start your presentation. And it's already 8 minutes gone, so start.
present. Is the screen visible?
Yes, it's visible now.
Okay. So, good afternoon, everyone. I am Sneha Rana. I am a PhD student from IIT Bombay and a TA for this course. So, today we will try to look at few problem-solving questions for the entire 1 week course. So, this will help you to solve the assignments for the week one. So, let's start with it. So basically, week one majorly. Just a minute, I guess someone has joined. So in this problem solving session, we will use the concepts that have been taught in all the videos of week one, and we will try to solve them. So the first problem is upon Ramachandran plot. So, as you know, Ramachandran plot is a very important discovery in the protein structure and protein chemistry. So, what Ramachandran plot tells you? So, it will tell you whether a particular protein structure, whether it is favorable or not, and what kind of a structure a protein will adapt depending upon its phi and psi value. So, phi and psi, these are the dihedral angles. So, basically, phi is a dihedral angle between C alpha, that is C alpha and N group, and psi is an angle between C alpha and carbonyl carbon group. So, calculating this 5 and psi values, you can actually predict the structure of a particular peptide, and you can also predict whether a particular peptide will adapt a particular conformation, a secondary structure or not. Because many of the plays in Ramachandran plots are forbidden because of the hindrance, a steric hindrance. So that can be calculated depending upon the phi and psi values. So here what I have done, I have given a set of phi and psi values. You need to tell me in the comment box whether first whether the structure is favorable or not, and secondly what is the possible secondary structure that a particular peptide will adapt. So here I have given you four different sets of values for 5 and psi for different peptides. Let's say these are A, B, C, D peptides. So you comment upon whether the first of all whether the structure is favorable or not depending upon the values, and which structure it will adapt. So you can write in the text box whether what structure, or you can unmute yourself and tell which kind of structure it will adapt, and whether this particular peptide will it have whether the structure, a secondary structure, is possible or not. So anyone wants to take the lead and comment upon this? Hello?
Hello.
Yeah, please go ahead.
Yeah, so I think the first one for psi minus 56 and the psi minus 45, that's going to be on the
I'm not able to hear you properly. Can you please keep the mic in a proper condition?
okay. Hello.
Yeah, yeah.
Okay, so I was saying for the the psi value minus 56 and this phi value minus 45,
Okay.
that falls on the lower left quadrant.
That will this structure be favorable or not? First comment upon that.
I think it's going to be favorable because it falls on the lower left quadrant, which is the accepted alpha helix. It's going to
Okay.
I think it's yeah I think it's going to be in the accepted alpha helix quadrant on the
Okay.
lower left quadrant. Yeah.
Okay. So you think it is possibly an alpha helix. Okay.
I think yeah.
This is anything. Definitely it's favorable. Okay. Uh, Anyone wants to try second? Minus 119 and 113. So you can use this plot to locate the region. So this is minus 5 region, this is plus 5 region, this is plus psi region, and this is minus psi region. So this will give you an idea at what particular location but this values will fall upon, and depending upon the location, you can decide whether it's a forbidden region or whether a particular structure is possible or not. So that is why I have given Ramachandran plot as a reference.
Hello.
Yeah, please go ahead.
Can I try again? Okay. So I think the second one I think is not acceptable. It's not favorable.
Okay, just
That
Yeah, can you please repeat what were you saying?
Okay. The second one, I think it is not going to be favorable because that falls
It's not going
on.
to be favorable. You
I think so. It's not favorable.
okay? And what is the reason for your thinking?
So it falls in the, you see, it falls in the lower right quadrant.
Lower right one. This you are telling?
Yeah. Yes. So that area is not acceptable, I think, according to Ramachandran rules.
Okay. But if you see, it's here is the
Hello?
phi value, okay, and the
Hello?
lower right region, here it is positive. So this is negative value. So it will be somewhere here. It won't be the lowest right quadrant. It will be somewhere in this. So if you want to rethink again and answer,
Okay. Let me check again.
because it's minus in the phi
Hello?
region. Yeah, please go ahead.
Uh, I think it will be in the second quadrant, that is in the top left one.
This one. Yeah, correct. So if
Yes.
you see the value 5 is minus 119, which is negative here, and psi is positive. So perfect. This is the quadrant. So you have identified the quadrant correctly. Now, what do you think in this quadrant? Is it will it be a favorable or it's a forbidden region?
Yeah, it's a favorable region for the beta sheets.
Yes, yeah, you are correct. It's the favorable region for the beta sheets.
And specifically, if you want to tell, it's for the parallel beta sheet.
Yes, you are right. So both the beta sheets goes in this particular region. So between the parallel and anti-parallel, there is a slight difference in the values of 5 and psi. Now the third one. Third one, anyone wants to try?
Not favorable.
Yeah, please go ahead. Someone was saying something.
It's not favorable because it comes in the third quadrant.
It's not favorable. So we will see 5 is plus positive value, which is this, and psi is negative, which is this region. Correct. So this comes under this quadrant, which is not favorable. So you are correct. This is not favorable. So definitely it will not have any possible structure. And what about this? So here both 5 and psi values are positive. So 5 is positive, which means it will be in this area. And psi is also positive. So this. So it will be in this quadrant. So is this area favorable or unfavorable for secondary structure adaptation?
Yeah, this is favorable for left-handed alpha
Yes,
helix.
Favorable.
correct. So any idea which structure it will take?
Alpha helix.
Yes, alpha
Yes.
helix.
Specifically, the left-handed alpha helix.
Yes, more precisely correct. This will be left-handed alpha helix. So this region is favorable for left-handed alpha helix here. So this is the value. It is for right-handed alpha helix. So I'll just show you this diagram. So this is just a classic diagram which will show what are the favorable regions, and the white region shows forbidden areas. So these are sterically not possible for a particular peptide sequence to adapt a secondary structure in these areas. So as we have discussed, so last one was showing both the values positive. So it is left-handed alpha helix. The first one, if we see, was both the values were in negative. So both the values were in negative, this region. So this quadrant. So it is right-handed alpha helix. Whereas if you see the second one, it is phi value is a minus and psi value is 113. It's positive. So it's beta sheets. So it is here. So it is beta sheet. So this is a very important discovery. You can just predict the structure, a secondary structure, depending upon a peptide sequence. If you have a particular sequence, you can just figure out which structure it can adopt. So we will move to second question. So this section here we will be talking about mutations and how it affect at the end a protein structure. So basically, we know that genes code for that genes codes for protein. Genes are transcribed into mRNA. mRNA is then converted into protein via translation mechanism. Okay. So now let's say there is a particular protein coding gene sequence. Okay. And so this is the gene sequence GAC. Now it is converted into GAT. Okay. So you see there is a change in the C to T, and there is another mutation in this particular gene sequence. AGC is converted to ATC. Okay, so my question is that will this mutation affect the protein structure? So you have to comment whether it will affect the protein structure or not. So I have also given,
AGC to
yeah, sorry,
AGC to ATC will affect the protein structure.
sorry. I was not able to hear you. Can you repeat it? Hello?
Sorry, my mistake. You can continue.
Yeah, you can repeat. I was not able to hear you. Okay, so this is
Write in the chat if you are saying something. You can write in the chat also. We'll see.
so this you can use as a reference to find whether these particular sequence, the change in this mutation, a point mutation, will this affect a protein structure or not? So this is a gene sequence. Remember that. And here it's an RNA, mRNA sequence, the codons, mRNA codons. So take your time and let me know.
Sneha, Divya has written in the chat that AGC to ATC will affect the protein structure. So,
Okay, AGC to ATC will affect the protein structure. Okay, fine. So that is option 2. Okay. And what about the first GAC to GAT? Will that affect the protein structure or not? Anyone wants to comment upon GAC to GAT? What will be the change happening if that particular sequence is changed?
yeah, Navneet has written GAC to GAT won't affect.
Won't affect. Okay. So why this? Okay, you guys are correct. So why GAC to GAT will not affect? We will see. So this is GAC. Okay, this is the mRNA CUG. Okay, and this is GAT. So mRNA CUA. So let's just see what protein it codes for. CUG and CUA. So let's see CUG. Okay, CUG and CUA. So you see both are coding for leucine. So the protein at the end is not changing. So it will not affect the structure of protein. Whereas if we see in case of another mutation, this is replaced by UAG, which is a stop codon. Okay. So now, because the stop codons has come in between, the peptide chain that will be generated will be truncated. So definitely, the protein structure will be affected by that mutation. So just to give you an idea that there are few different kinds of point mutations occurring and in the body. So silent mutation. So the first mutation which we discussed was a silent mutation. There was no change in the amino acid was happening. So even if there was a slight change, there was a change in the gene sequence, there was no change in the amino acid. So that is why your protein structure will not be affected. Then the another mutation that we discussed was nonsense mutation because it was replaced by a stop codon, and that's why it will generate a truncated protein. And then there are other missense mutation. Here, what will happen? Amino acid will be replaced by another kind of amino acid. So missense mutation are of two types. Okay. So change in a gene sequence, what will it lead to? Suppose there is a particular sequence that is coding for lysine. Now, due to point mutation, now it is coding into arginine. But if you recall that lysine and arginine have a similar physicochemical properties. Both are charged polar side chains. Both have charged polar side chains. So that is why the protein structure will not be that much affected. It will still be able to fold in a particular manner because of the similar properties. Now there is also a non-conservative missense mutation. So here what happens? A particular change in a gene sequence led to change in amino acid sequence. However, the change is in a different kind of amino acid. You see here lysine is changed to alanine. Now alanine is a non-polar aliphatic amino acid and lysine is a charged polar. So you see there is vast difference in the properties of lysine and alanine. So definitely if lysine is replaced by alanine in a protein, it will not be able to serve the function of lysine, and that's why a protein structure will be hampered. So these are the different kinds of mutation that happens, and this could also lead to various diseased conditions. So silent mutation, there won't be any diseased condition in case of missense mutation, especially non-conservative missense mutation. These lead to various disease. So, anyone have an idea, like know some disease which is caused by point mutation, which leads to change in the amino acid sequence? Some example, if anyone has. So, you guys know about sickle cell anemia?
Yes, yes.
Yeah. So it happens just by 1 point mutation. Okay. So in that one, due to 1 point mutation, there is change in the amino acid. That's why the shape of the RBCs is become sickle cell. So now we will move to another question. So this is the third question. So let me first read the question. Ionization state of amino acids changes with varying pH. Okay, given below is a representation for change in the concentration of ionization states of an amino acid across pH. So this is a graph of pH versus concentration. Okay. So figure shows three different colored lines for representing the forms of amino acid. Based on the information provided, answer the following question. So you can see here, so this is a red line. So here the concentration is decreasing, okay, with the pH. Here it is increasing then it is stabilizing and again decreasing. So you have to tell the state. So whether the protein or sorry, whether the amino acid it is in a protonated form, deprotonated form, or a zwitterionic form. So zwitterion is basically it will have both positive and negative charge. And further you have there is a second extra additional question in this. So determine the pH at which the following two conditions. First is when the concentration of protonated form is equal to that of zwitterionic form, and when the concentration of deprotonated form is equal to that of zwitterionic form. So carefully observe this graph and try to think. So if you see in this red line, okay, with increasing pH, this particular ion form it is decreasing, so it is giving you some hint.
I think one is protonate form,
Correct.
and two is zwitterionic form,
Is deprotonated. Okay, I think you should think again.
and
3 is
three is deprotonated.
okay, 2 and 3 both are deprotonated.
No, three deprotonated.
3 is
And
deprotonated
yes, and two zwitterionic.
and 2 is zwitterionic. Okay,
Yes.
yeah, so you are correct. One, the red line is protonated. So if you see with increasing pH, it is the concentration is decreasing. So you can figure out that the protonated form, because of the increasing pH, it is decreasing. Okay, so that is what it will happen. So after once a proton is lost, and you can see the pH is increasing. Okay, so the first proton will be removed first. After that, the second proton will be removed from the carbonyl carbon. So which will lead into the formation of zwitterion. Okay, I have a figure, but so that is why this represents a zwitterionic form. And even if after that you keep on increasing the pH, okay, so it will lead to a deprotonated form. So finally, even the last H will be removed. And the second question is when the concentration of protonated form is equal to the zwitterionic form. So this you can figure out that here, uh, so this concentration of protonated is decreasing, right? So right at this point, the concentration of deprotonated form is also increasing. Okay. So at this particular point, the concentration of protonated and zwitterionic form will be equal. And after that, with increase in pH, the protonated form will be still further decrease, and the zwitterionic form will increase. And here it will be in a steady state. So here the concentration of zwitterionic form will be uh, stable. And after that, here the concentration of zwitterionic form will decrease, and eventually the concentration of deprotonated form will increase. So this is the concentration. pH 10 is the concentration where the concentration of zwitterion and deprotonated form will be equivalent. So if you see here, almost 50% has converted to the deprotonated form, and here it is almost 50% is of deprotonated form and 50% is of zwitterionic form. So anyone has any doubts here? Should I continue to next question? Okay, so I'll go to the next question here. So this question is from Anfinson's experiment. So Anfinson's was discovery majorly talks about that the a protein structure, a native structure that a protein adopts, it is inherent in its uh, amino acid sequence. So that is what was proven by Enfinson, right? So what he did an experiment with RNSA, right? So you would have read about this state. So he added urea and then the protein was denatured, and when he removed the urea, again the protein was able to fold in its native state. So here is that experiment. So we will first read the question. And Vincent performed several experiments which demonstrated that the information for forming the three-dimensional active form of a protein is present within its primary structure, which is amino acid sequence. Given here is a schematic representation of Anfinson's experiment showing four different experiments with transition between three different forms of protein. So these are three different forms. This is a native functional form, this is denatured form, and this is scrambled form. So depending upon the given information, answer the following question. So experiment 4. So you can see here this is experiment 4. So this is from scrambled to native state. Okay. So this particular experiment involves dialysis of urea and addition of traces of mercaptoethanol. Now, what would have happened if mercaptoethanol was not added at all, but by mistake urea was added? So, instead of adding traces of mercaptoethanol, you are adding urea again. Okay, and remember that you are dialyzing urea, which means you are removing urea, but now you are adding urea again. So, what will happen to the protein? So, So these are the four options. Protein would have transformed back to denatured state, that is this, in the presence of urea. Second option is protein would be inactive with scrambled disulfide, which is this state. Third option is protein would resume activity with normal disulfides. Okay, which means that it will resume the activity, so it will be in a native state. And fourth one is protein would have normal disulfides but no activity, so it will have the disulfide bonds but there won't be any activity. So this is a thinking question. Think about it. From this state to this state, scramble state to native state, you have to remove urea, but by mistakenly you are adding urea, so what will happen?
Hello?
Anyone wants to comment?
So Sneha, Devya has written three protein resumes activity with normal disulfides. Like
Protein would
option two.
option 2 or option 3?
No, sorry. Divya has written option three. Ankit Mukherjee has written option two.
Okay. So one is saying option 3, which is protein would resume. Just a minute, I think there is one. Continue. Yeah. So option 2 is protein would be inactive with scrambled disulfide, which is this form. And option 3 is protein would resume activity with normal disulfides. So it will resume activity, meaning it will be in a functional state, which is native state. But if you see in experiment 4, okay, you are dialyzing urea, but you have by mistakenly added urea. So if urea is already added, how will it go into a native state? Because to go in the native state, you need to remove the denaturant, which is urea. So um, protein would resume activity with normal disulfide is not correct. So second guess, which was protein would be inactive with scrambled disulfide, which is this state. So you are dialyzing here, but again the urea, the same amount of urea has been added. By mistakenly you have added urea. So it is not going into any further state or denatured state. Besides, you have not added ethanol also. So it will not happen, anything will not happen to the disulfide bonds. So it will remain in this particular scramble state only. It will not go in a native state because urea is not removed, and it will not go into denatured state because mercaptoethanol is no, it's not there. There is nothing which can break the disulfide bond. Okay. So that's why option 2 is correct. Protein would be inactive with scrambled disulfide, which is it will be in the same C state. I hope this question is clear now. Should we move to the next question? Okay, so I'll move to the next question. So this is also okay. I have already given the solution here. So anyway, we will discuss this. So suppose you are very hungry and you made an omelette. Okay. Suddenly your friend came and you decide to eat pizza. So as per the Anfinsen experiment, if you remove the denaturing agent, so here the denaturing agent is heat. Okay. If you remove that, then it should fold again in the native state, right? So why does it not happen in case of egg protein? Why does it does not form again the egg? Why omelette does not form again egg? So I've forgotten to remove the solution. So I'll just discuss why it does not. So as you know that native state of the protein is the most favorable, but most thermodynamically favorable state of the protein. So if you can see here, so here is the free energy. The delta G value is negative, and here the native state is most favorable, and that is the state that protein takes. That is the three-dimensional state that protein takes. So here the delta G negative value will only tell you about whether a particular reaction will occur or not. Okay. So omelette will the native state of a protein, egg protein, it will be formed. It's the thermodynamically most favorable form. Okay. So thermodynamically, it will tell you that that form is possible to occur. That reaction will happen. It's a spontaneous reaction, so definitely it will occur with delta G value negative. However, it does not provide any information regarding the kinetics. So kinetics will not be dependent upon the delta G value. Okay. So here in case of Anfinsen experiment, even kinetically it was a favorable reaction as well as um, thermodynamically it was favorable. So here the reaction is not kinetically favorable. Uh, so that will not tell you about the time, how long it will take to finish that particular reaction. So to again form an egg protein from a denatured egg, just a minute. So uh, the formation of egg protein again from a denatured egg is not possible because the delta G value will not tell you about whether how fast a reaction will occur or not. Delta G will only tell you that this particular state is spontaneously and thermodynamically possible. Okay. So this is just a fun question which I kept to make you aware that even if in case of Anfinson's experiment, while he removed urea, it was able to take the native state again. But in case of many proteins, this will not happen. Why? Because of the kinetics. Because delta G is negative, so definitely that protein will try to take a native state, but it will take a very long time. So sometimes when you are doing protein refolding experiments, adding particular enzymes helps in increasing in reaching the refolded state faster. So because enzyme, what it increases the, I mean, it increases the speed of the reaction. So do not confuse that every time you remove the denatured, it will it will form the native protein always. So this is another question. So here, Nana wants to study difference in the protein expression of diabetic patients using serum samples. Okay, so she wants to study protein expression in the diabetic patient. So she's comparing a control group versus a diabetic group and using SDS to see what is the difference. But if you see, so this is the gel image of her experiment. Okay, after sample processing. So comment on why she cannot distinguish between control and diabetic group. So if you can see here, there's no difference. If you see, it's almost similar. So she's getting a big bands in across all the gels. These are big bands, and she's not getting the proteins different in the proteins. So any reason for this? So, okay, so if you remember, in one of the lecture it was discussed. So whenever you are doing a proteomic study, one of the major important step is to remove the abundant protein. So in serum and plasma samples of humans, um, globulins, albumins, they are very high in concentration. They are highly abundant protein. So if you do not remove that, and you do your analysis, you will only get those protein bands, and the protein, the low abundant proteins which you are interested in, so which could be changing in case of your diabetic samples or any disease sample, you won't be able to identify those proteins. So that is why it is very important that you remove this high abundant proteins from your samples before doing any proteomics experiment. So generally, affinity chromatography is carried out to remove these high abundant proteins like albumins and globulins. So this is a very crucial step. If you forgot to do that, you will the whole entire process of proteomics will be uh, you will not get that particular result you want. You won't be able to see the difference in other low abundant proteins, which could be the proteins of your interest. Anyone has any question regarding this or any doubt? Should I move to other question then? I'll move to this another question. So this is an experiment about 2D dyes. So here Sunita has performed 2 dyes to study differential protein expression in Huntington's disease. So she has labeled control group with Cy3 dye and disease group with Cy5 dye. So here what you have to do, you have to calculate what is the fold change in the known proteins A, B, C, D and comment which of the proteins are differentially expressed. Also identify visually which proteins are potential biomarker for Huntington's disease. So dyes is a very useful gel-based technology wherein you can study differential protein expression. So here first of all you separate the proteins based upon isoelectric focusing, and then the second dimension, in that you are adding these different kinds of dyes. So you are labeling your control sample with a different kind of dye, and um, your treated or disease sample with another kind of dye. Okay, and then you will see uh, the fluorescence at both the different kinds of wavelength particular to that particular dye, and see the expression. So suppose if a particular protein is highly expressed in case of healthy, and its expression is decreased in case of a disease condition, so like in case of E. So here you can see that this protein is highly expressed, and here it has decreased. So that way it will be more fluorescent in case of when you are seeing with Cy3 label dye, and it will be decreased in case of disease group. So in that manner you can identify the differential expression of protein, whether a particular protein is down regulated or up regulated, comparing to the control group. Okay, so this is a very nice technology to study the differential expression in case of any disease or uh, healthy versus diseased, or any treatment, whether some drug treatment if you are giving, and then trying to see the expression of different proteins. So this is, I guess, this is simple. So anyone wants to just say which proteins are down regulated? So one I have already discussed is E. This is down regulated. You are seeing its expression has decreased in this Cy5 label. And Cy5 is a disease group, so it is down regulated. Another proteins, anyone can comment that which one is down regulated and which one is uh, up regulated.
Okay, so Divya has written B and C are upregulated. Ankit has written D is downregulated and B is upregulated.
It's down regulated. Okay. So here I
And
have also given the
Divya
value.
has written again G also upregulated.
Yes, correct. G is upregulated. B is also upregulated. B is also upregulated. So we'll go with A, B, C, D. So A, if you see, A there is not much change. So here I have also given the values. B is upregulated. C is uh, C is similar. So there is not much difference in the protein expression. H is also similar. D is all D if you see. So here the values, depending upon the values, it is down regulated. So it's just the error in the figure. So it's down regulated. The values are decreasing in case of sci-fi. So in that manner, you can figure out using this method whether a particular protein is upregulated and downregulated, and that could act as a potential biomarker for any disease which you are interested in studying. So this is the end of the presentation. If you have any doubts, you can ask. So here you need to calculate the fold change to know which are the proteins which are upregulated and the down regulated. Okay. So in this manner, DIGE is used to find the difference in the protein expression between two different groups or different groups. So anyone has any doubts in any of the question or during the week one lecture video, you can discuss now.
Abdul has written, can you explain the slide 8?
This one, slide 8. This, na? Okay, so here I have shown the diagram of free energy. This is entropy. So this is the funnel. So this is giving you an idea of which is the most probable native state a protein can adapt depending upon the free energy. So if you see here, the free energy is decreasing. So mostly the lowest free energy state is the most probable stable conformation a protein can adapt. Okay, so that is why here the delta G value is highly negative, and that is why it's a most thermodynamically probable form of a protein, uh, that a probable 3D conformation of a protein that it can adapt. But as I said, it will, the free energy or the delta Z value will only tell you about whether this particular protein will go into a native state or not. So definitely it will go into a native state because it is thermodynamically more favorable. But as I discussed, it will not give you an idea about kinetics, meaning how fast or how slow a reaction will occur. It will not tell you that. So that is why in case of egg protein, when the protein is already denatured, you cannot make it again egg while you remove the heating condition, because although the reaction is possible, kinetically it is not possible. It will take years to make that egg again. So kinetically it is not possible, and that is why in most cases you will not see that even if you remove the denaturants, it will not form the it will not go into the native state again. So this is slightly paradox to Alfinson's experiment. Alfinson proved that while you remove the denaturing agent, it will take the native state, which is correct because it has the amino acid sequence, it has the primary structure, and the information to take which state a 3D structure, it is already there in that uh, primary structure of the protein. But it will not it will not happen in all kinds of enzyme or all kinds of proteins, although it is possible, but it will not occur. That is what I want to say. That's why I included this question. If you don't have any doubts, should I stop sharing? Yeah, stop sharing now. I think that no one has any doubts. So
So thank you everyone. So we are floating the Google form every week. So if you have any doubt regarding the lectures in any week, so you can form the Google form. So in this live interaction session, we will try to solve your queries. So you can fill that Google form. So thank you everyone.
thank you everyone.